infectious diseases. They are the first line of defense, but at the same time, vaccines came into the picture in last 2 years since the COVID pandemic has happened. And everybody was looking at the vaccine, and there were doubts were expressed, like Dr. Nivedita talked, whether we can make those vaccines or not, and whether it can come on the time or not. That was an issue which was flagged by everybody. But to our pleasure, I think I'm glad that vaccine came in just within a year's time. The vaccine was developed, and we started vaccinating the people. So today in India, almost 70% people are at least taken single vaccine dose. 56% have taken both the doses, and 1.3% are taken even the booster dose. So a lot of people have been vaccinated. So this happened because of the efforts of our scientists who have developed these products. I think two of the pioneering work who have done in this area are here. Dr. Kapil Maithal, who is the president of Zydus Cadila, particularly vaccine division, and now I think Zydus Cadila Healthcare has gone. Now Zydus Zydus Pharmaceuticals. Yeah, and I think he has got a honor of introducing the first DNA vaccine in the world, which was even talked about by our Prime Minister in the UN Assembly session. Second person we have is Dr. Sanjay Singh, who is the CEO of Gennova Biopharma Private Limited, Gennova, which is the subsidiary of the MQR, right? He has worked with the Gennova since 2006. Before that, he was with the National Institute of Health, USA, where he headed the antigen research section. He completed his graduation and post-graduation in science from Lucknow University and postgraduate from the Central Drug Research Institute, Lucknow. So I think we have got a great panel who can really deliver what is required for the today's session, further details. So I think first question, can I ask to Dr. Kapil? First question, I think Zydus Cadila has been one of the pioneers in the vaccine department. A lot of vaccines have been developed and marketed by the Zydus so far. Can you give your insight on the process of vaccine development from delivery, design to delivery? Dr. Kapil, please.
Thank you, Mr. Belapure, and thank you for the organizers for having such an apt meeting or a summit discussing about vaccines. And so coming to the question of Mr. Belapure, I think vaccine design and development is a very interesting field and very different from the classical pharmaceutical development. So one basically starts at looking at the pathogen which is responsible for a particular disease. Usually infectious diseases are the ones which are targeted by vaccines, even though now there are vaccines for other non-communicable diseases which are in research and development. So once a pathogen has been identified, then one needs to understand the host pathogen interaction and identify what stage of the pathogen one needs to target or develop an immune response to eliminate the pathogen or to let it be attenuated to an extent where the response is very limited and the person doesn't get the disease per se. And once that is done, then you also need to understand the kind of serotypes or genotypes of the pathogen. There are a number of diseases like caused by, like, say, Streptococcus pneumonia. There are more than 100 serotypes which are prevalent. And one needs to identify what are the serotypes which are prevalent in a particular region or the major serotypes against which a vaccine or a multivalent vaccine needs to be developed. And having identified the antigen, one then needs to understand what kind of an immune response is required. Whether one is looking at a humoral response or a cell-mediated immune response or both responses. In humoral response, if one is looking at systemic response or a mucosal response or both responses. So I think that's where it becomes very interesting to understand what kind of excipients or the antigen design needs to be done, or what kind of adjuvants needs to be built in. Having understood that, then basically the vaccine development starts. Now here I'd like to mention that there are different platforms of vaccine development. The classical ones being the live attenuated viral vaccines, something like what we see in measles or rubella vaccines. Then we have inactivated viral vaccines, something like hepatitis A or seasonal flu vaccines. Then there are subunit vaccines, which are recombinant vaccines, like our hepatitis B or hepatitis C. The recent vaccine for malaria, which got approved in 2015 from GSK, is again a recombinant protein vaccine. Then one also have vaccines with polysaccharides, polysaccharide conjugate vaccines, toxoids like tetanus toxoid, or diphtheria toxoid. Then one can also have the new platforms which are coming up in terms of nucleic acid vaccines, the DNA vaccines and the mRNA vaccines. They specifically came up during the COVID time. And then non-replicating viral vaccines, something like what we have seen in the case of Covishield or Sputnik, which are based on adeno platforms. And these are again new platforms which came up during the COVID times. So having understood the different kind of platforms, the different kind of antigens present in a particular pathogen, that is when the immunogen design happens. And once the immunogen is developed, then the standard process development or follows before one goes into the conventional regulatory toxicology studies in animals and then clinical trials. And it becomes important in the clinical trials specifically if one does not have a correlate of protection established. In phase 3, one needs to do a large efficacy trial, as we have seen in the case of COVID vaccine, the initial COVID vaccines, where a large clinical trials were done to establish the safety as well as the protective efficacy of the vaccines. So that is the development part. You talked about the delivery. I think delivery becomes very important in terms of logistics, cold chains, whether one is looking at a single dose presentation, if it is for a private market, or a multi-dose presentation, if one is looking at the public market. In the case of COVID vaccines, most of the vaccines are multi-dose vaccine because that is ease of logistics and supply chain management. And that is how the entire path of vaccine development and delivery happens.
Kapil, one supplementary question. How do we decide which platform to use, whether to DNA based, attenuated? So how that process goes on?
So that's a very interesting question. And as you would see in the case of COVID vaccine, it's not a single platform on which these vaccines have been developed. And in fact, many of these platforms have been successful. At the same time, there have been a lot of platforms where the vaccines have failed and not moved forward. So one can develop on different platforms. The classical platforms I already mentioned to you. It depends on what kind of a pathogen, as I mentioned, is to be targeted. In COVID, it was new to all of us. And so the nucleic acid platforms were used. We have Covaxin, which is an inactivated viral vaccine. We have Covishield, as I mentioned, and Sputnik, which are on non-replicating viral vector platform. So various platforms can be tested and tried. It depends on what one is looking at, how rapidly it can be developed, what is the logistic ease of developing these platforms, as well as the cold chain requirements. I think that is very important.
Thank you. Coming back to Dr. Sanjay Singh, Gennova has been in the forefront of the biopharmaceuticals and vaccine manufacturing in India. What, according to you, are the best practices that industry can adopt to overcome challenges in manufacturing and development of biopharmaceuticals as well as vaccines? Can you throw some light on this?
Thank you, sir. Either in pharma sector or any sector, if you ask that, can you offer the best possible product? in healthcare, it is much more required as compared to anything because it touches the human life. So COVID was such a big problem because we were more in the research of developing different technologies, adjuvant delivery system in the vaccine for last 10 years with different national and global collaborations. But when COVID came, mRNA platform, we were working on the personalized cancer vaccine program, and we put this together. But the speed and the requirement was needed for this. We tried our best and following all the possible rules because mRNA vaccine coming from India, global eyes are there. But with mRNA vaccine, there is a one deployability problem. It required minus 70-degree ultra temperature. That would have not been deployable in India and most of the world. So we have gone ahead and we innovated that part also. As I said in my opening remark, you have to innovate by which your product would be the best possible in the class and deployable and then affordable. So certainly regulatory frameworks have been changed, has been looked into. They have worked very hard. But for the platform like mRNA, it was never heard of in India. So our expert was sitting at either at office of RCGM or of office at DCGI. It was not a simple journey for us, not for the regulators. But I think because of this is such a complex situation, we needed all kind of platforms. So depending upon where the ease was the highest, those platforms move forward. And where the ease was, because regulator also have to ensure themselves 10 times before giving the approval. So this is the way each vaccine has gone. Now, taking forward what Dr. Nivedita Gupta has said, that this is a big learning session for us. Learning session in the sense, there are two types of situations are there. We used to keep a huge army. There is no fight every day. But if there is a war open, then you should have a situation you can put a deterrent, you can put the army, you are prepared. So something like these kind of pandemic are same. So nobody was ready for this kind of pandemic, and the way that losses has happened all across the world, you know it. So what we learned from is combination of yes, the ease, the government support, the industrial support, working together with policymakers, regulators, Ministry of Health across testing, those things has given us a lot of lessons. So we have fast tracked our development. But at the same time, there are another kind of challenges came. For the most modern technology, for the standard technologies, things are available in the country. But for most modern technologies like mRNA, we didn't have a single thing available into it. So can we look into create a situation which could be stockpiling, that if next pandemic or next something comes, we are in position to do it. And not only through our internal recruitment, but we can talk to the partner, we can talk to different kind of manufacturer, and government should also look into it. Because we have seen somehow, because of price pressure or L1 pressure or those kind of regulations for the government also, the people have gone back to China to take all the API. So the whole API business was gone. Now the government is putting back PIL. So I'm saying your question you asked for the manufacturing and quality when concerned, my thing is we have to prepare to deliver the best possible product in its class, not only for India, globally. For that, companies has to get ready, companies has to innovate, companies has to come out of this generic mental framework, and we are trying to do that, not only saying we are trying to do that, but equally regulators has to be ready to review those kind of situations, and they should be available guidance. So these are my learning from it, and we have to build on for the future from here. Another point is that we were knowing that the kind of virus it is going to go under huge mutations. And mRNA possibility is that where you can within no time you can convert it because it is absolutely synthetic platform. So we have submitted our paper. We are waiting for the approval for Omicron clinical trial, and hopefully those kind of modern solutions are required. So what is required for the nation, what is required for the population, and best in the class, we as industry we have to move into that form. And also, because we have to also take the value chain forward, because low-hanging fruits are very easy. So, and the regulator also have to go forward. So this is my comment on it.
Thank you, Dr. Sanjay Singh. I think COVID really taught us how can we work together collaboratively. Government also was very responsive this time, and they, knowing the fears which are getting spread across, I think government also helped in the collaborating and getting the things faster. Do you feel that the regulatory systems help to get it a faster mode? Did it help this time?
Yes, regulatory system is very open, and they have, I would like to say, starting from the minister, starting from the office of Niti Aayog, or starting from the secretary office or the DCGI office, they were available 24/7. So they have done, not only we have done unprecedented work, the regulatory authority, the government has also done the unprecedented work. The question here is that if we have become to the best, still we have to walk through that. Still there are a few things where we have to move forward. And always there is a huge scope of improvement. And I feel that true is for us, that true is for regulatory, because again, a product in the market. If we don't deliver best of class of the product, I would like to say, then the generic business will become fiat and investor. You will just go away from the market because other competitor products are available.
Thank you. Thank you, Dr. Sanjay Singh, for explaining. Kapil, another question for you again. What kind of the challenges you face? Because you developed the DNA-based vaccine first time, like they developed mRNA, you developed the DNA-based. So what were the challenges in the all across, means development, regulatory challenges, manufacturing challenges, technology transfers, supply chain? Can you throw some light on this?
Yeah, definitely. I think when we started thinking about developing a vaccine for COVID, which was somewhere in January of 2020, when there was in fact no case reported in India, but the way it was spreading in China, we knew that it's just a matter of time before this can become a global pandemic. And we started evaluating different platforms. So DNA vaccine platform was something which had been tested in past, but there was no human vaccine available. And two of the earlier coronavirus infections were MERS and SARS-1. There were clinical trials which were very successful, but they did not go to the phase 3 clinical trial. They basically culminated at phase 2 clinical trial. And probable reason was that these diseases were self-limiting, restricted to certain territories, and they did not need to go to a large phase 3 clinical trial, which has its own requirements, both in terms of population as well as monetary requirements. So that was one of the platforms we thought. We also evaluated other subunit vaccine platforms. And I think, as Dr. Sanjay also mentioned, one needs to look at the ease of development as well as how it is going to be used in the population. And looking at our country and other LMICs where these vaccines would be required the most, we thought of working on a DNA vaccine platform. There were a lot of benefits of that. It was very safe, it is non-infectious, it poses no anti-vector immunity. We knew that would be a lot of mutations happening. As the immune pressure would mount on the virus, it is bound to develop escape mutants, and that is what we have seen now. So this is again a plug and play technology where we can develop newer candidates as soon as possible. As Dr. Sanjay mentioned, in an mRNA, he already is waiting for a clinical trial to start for the Omicron candidate, because that's the beauty of nucleic acid platforms. Even we have an Omicron candidate ready, which is already being tested in animals right now. So one can move very rapidly on this platform. So that was another reason we thought we can work on it. Requirement of adjuvants, which we know is mostly imported in a country, can become a challenge in long term. So that was one thing which we thought in a DNA vaccine, and adjuvants would not be required, would be very easy. And unlike many of the vaccines, in fact, unlike any other vaccine platform, DNA platform provides a very safe vaccine in terms of, even in the terms of stability. and safety. And the candidate can be stored for at 25 degrees for a few months, even at 37 degrees Celsius. So it becomes very important again in countries where cold chain requirements can lead to a lot of vaccine wastage and all that. So having thought through that, then we started looking at, as I mentioned in the first question of yours, about how to do a vaccine design. And that is when we had to select the target gene, the sequence, the promoter, the signal sequence, which will help in a better uptake of the plasmid DNA by the cell and elicit an immune response. Again, the selection of the cell substrate was very important. What kind of a cell substrate is required, which can be used to propagate the plasmid DNA. Also looking at the kind of vector, because that becomes very important from a safety perspective. And you mentioned about the regulatory. I think we were very skeptical ourselves to ensure the safety of this platform. And we did a lot of studies to ensure the safety of this candidate, starting from early preclinical and in the clinical development. So once the vector was selected, then there was not much guidance available in the literature. So we had to look at what kind of an analytical methods would be required, what kind of animal species would be required to test these vaccines. And in fact, at that point of time, we tested this vaccine in numerous animal species, number of species of mice, rats, rabbits, hamsters, guinea pigs, even dogs, and also in non-human primates. So fortunately, we have a world-class animal house, and all these studies could be done at a very rapid pace. So we did this, identified a suitable animal species in which the different candidates could be tested, and then we went ahead with the process development. Now, most of the people on this conference would know that there are simple kits which are used for purifying plasmid DNA at a lab scale. The standard molecular biology labs have that. But now we had to develop a process which can be scaled up, is commercially viable, and that's again a new challenge we started, which fortunately we were able to develop a simple process. And at the same time, we started investing in developing a facility because we knew once the clinical trials would be over, if the vaccine is found to be safe and effective, we would need to have commercial facility to be ready. So that was a huge risk, and the organization took that challenge in investing on development of this facility, and that too during the first and the second wave of COVID, when getting the infrastructure, equipments was one of the major challenge. And subsequently, one of the major challenge in DNA vaccine is vaccine delivery. And so we started evaluating the different devices, what could be a suitable route for delivering of the plasmid DNA. And mostly the clinical trials which were happening across the globe earlier for DNA vaccines used electroporators. And we realized that would be very cumbersome. You would need a power source and probably an additional impulse or a pulse of this electric current to be given to an individual after the normal injection would probably defeat the purpose of ease of delivery. And that point of time, we identified a device called a Pharma, which is a needle-free injection system. And with this, we moved forward, and fortunately, we got very good results in terms of both humoral as well as cell-mediated immune response for the vaccine candidate. Regarding the regulatory, I think they were also learning as we were moving forward. Since we were one of the early starters on vaccine development, at that time we did feel that the pace was slow. But I think once the speed was picked up somewhere around April, May, the regulators also started giving a lot of waivers. They started supporting us. They were always there to advise on what should be done to expedite the program. And there were tremendous support from everyone. As Dr. Sanjay mentioned, from NITI Aayog, from PMO's office, from ICMR, from DBT, from DCGI's office. I think everyone was there, available on phone, guiding us and taking the development in a rapid manner. So I think that was the journey, how we went ahead on the development of vaccine before we got the emergency use authorization last year.
Dr. Sanjay ji, you want to add something in this? You have to unmute yourself.
Sir, I would like to say that yes, we have done a great work as a pharmacy of the world, but in terms of revenue, the percentage is much more lower in terms of volume. And I think with COVID, it has been shown and helped by the government that we can do things much better if we come together collaboratively for solve this problem. Not only for India, but we can do this job globally. So somehow now we have to take one step up or few step up where we have to, government also has to give some incentives or support system for innovation. Because last IPA was an innovation, and once the innovation comes, the company has to also take their neck out to do the innovation. Because if this is a very, as Dr. Kapil said, it is a very tough, difficult, and journey for us who has gone on innovative platforms. But certainly, we are also very cognizant of this. It is going to pay huge dividends in time to come. So, in that scenario, I will say that IPA, from the IPA mandate point of view, from manufacturer point of view, from the research point of view, from institution point of view, from the policy making point of view, we have to take this value chain forward. If we are not taking value chain forward, then a lot of competition is going to come where low-hanging fruit pickers are many more as compared to the innovation side. In one of the meetings, I said that yes, we can buy the aircraft, we can buy these things, but nobody is going to give you the 5th generation stealth bomber. So nobody is going to give us the mRNA technology here. So we have to develop our mRNA technology by our own, and I think government has listened us very carefully, and I'm very thankful that when DBT started, DBT supported us the seed money of around 125 crores. So that culture has started, but this culture has to be kept on, and by some incentive, by some support mechanism, by industry boldness, we have to continue this. This is just a start. We have to continue because we have a huge population, and to meet the need of this huge population is not an easy task. That is the only possible by innovation. So before that, if you see, there was our wired cables telephone system was there, but now the technology has gone into 36-channel telephone. Everybody can have the telephone. The same way, our benefits from the medicine has to reach to everyone. And for that, only way is the technology, high in technology and high in innovation. These are my closing comments, sir.
Thank you. Sanjay ji and Kapil, we have a very interesting session actually. We talked about the vaccines, everything, development, what are the challenges and how it was. What do you feel that, what are the things we should, he talked about some of the things, should be carried forward so that it becomes a norm instead of some of the shortcuts what we have taken today? Do you feel any regulatory reforms are required which will sustain us for longer time than currently what we have?
I think COVID has really taught us how things can move forward if it is taken in the right spirit. I think initial vaccines which were developed, even the traditional vaccines, they used to take somewhere around 5 to 8 years. And in the case of COVID, even the novel platforms, I think the first vaccines which came up were from Moderna and Pfizer, which were mRNA platforms, followed by AstraZeneca's adenoviral vector platform. These were all novel platforms. And still, the regulators were able to assess and do a risk-benefit assessment and get these vaccines on the market. Obviously, there would be learning, and as we have seen in some of these platforms, there had been adverse reporting over a period of time. But I think still the benefit had been a great advantage to the humankind. So I think with this learning, moving forward, I think what would be required is to expedite the development. A single window platform to have a faster clearances would be required. I think the support which was provided in terms of funding by different agencies, we also got grants from Department of Biotechnology to develop our vaccines. I think that would be very important. And regulators, as they did in COVID, have to be a support function. They need to be guiding the manufacturers and developers on the specific paths how to take this forward. In fact, in a lot of meetings, there were a lot of pointed questions which were asked, which helped us to do many other studies, which actually established the safety and effectiveness of these platforms. I think that is how the next, moving forward for the next generation vaccines, it needs to be done like that. India has shown that it can innovate. Now we have to take it to the next level. And COVID is one of the pandemics. There would be a number of other such infectious diseases which would be coming in near future.
Thank you. Sanjay ji, anything from your side on this?
I'm a very strong supporter of that all decisions related to drug development, drug manufacturing, drug delivery, drug pricing regulatory should be in one window, because still there is a lot of confusion on the policy side. So that part is, as a scientist, as an innovator, as an industrialist, I will strongly recommend, because government should take this decision as early as possible. Because this is our sunrise industry, and it has a huge opportunity to serve the nation and serve the world. So this is my first recommendation. The second recommendation will be that somehow government should be in supportive mode in terms of hardcore innovation. Hardcore innovation, it means if we see what kind of losses we control other than losses of humankind, our economy was lost around $1 trillion plus. If we have been not prepared in the first round, we made the preparation, how we are going to put the preparation for the second and third time, just like what we have disaster management thing. Can we have the pandemic management thing? Can we keep on looking on these things, how we can protect our economy, how we can protect our people. So from the policy side, this is the second my recommendation will be there. It should not be the post-COVID gone. It should be pandemic preparedness. Policy teams should be within the government at very high level, who should be put together a lesson learned and moving forward. So these are my two recommendations for the IPA.